Okay, so good morning, everybody, and great that you're still holding there and coming to this lectures on this reproductive immunology course. So what I want to touch with you today is associated to the subject of immune modulation. Yeah? What are the factors, who are the players that are maintaining this delicate balance at the implantation site between inflammation, no inflammation, rejection, tolerance, and so on. Again, that is a subject that is very difficult to cover in 1 hour, so I will try to go to general aspects, and I will focus with the macrophages as an example of the immune regulation. So we discussed already throughout the different lectures that pregnancy is characterized by the existence of a specific cellular and molecular mechanism that regulate the landscape within the implantation site that compromise or that maintains this balance by leading to the control of tolerance as well as protection. And we talk also about that pregnancy is in a unique immunological condition because it has all the components, all the immunological cells present at the implantation site. And I think you already have heard about many of these immune cells. We talk a little about dendritic cells. It was touched also about the NK cells and so on. So I want to focus today a little more about the macrophages, and we'll talk a little more about this. All of them, as we mentioned in the different lectures, are unique because they acquire, once they reach the implantation site, they acquire a unique phenotype that is present only in the uterus and is present only during the pregnancy. We also talk about antigen presentation. I'm not going to repeat, just to remind you that the antigen presentation that is occurring during pregnancy is different than the graft versus host. It's a very small process of antigen release that starts even at the time of conception with the sperm, proteins of the sperm. Then the combination of the oocyte and the sperm start producing unique proteins that are associated with the blastocyst. And the blastocyst in very early stages, already is releasing a lot of factors, including ACG, cytokines, growth factors, and all of the and other proteins that even we don't know. And all those proteins are reaching the maternal circulation, are reaching the uterus, and are taken by the immune cells of the mother, especially dendritic cells, and the process of tolerance initiated at that time. But of course, that is a tolerance associated for T cells, for T regulatory cells, who was discussed by Professor Joaquin. But there is also a tolerance that is happening at the implantation site to the innate immune system. Believe it or not, the tolerance is not only about the adaptive immune system. The tolerance also we refer about the innate immune system. Is a different type of tolerance, is more a regulatory tolerance that is happening at the implantation site. No suppression, is a regulation, because you need to maintain a strong innate as well as adaptive immune system in order to protect against infections. We will finish that the day of tomorrow. So how this immune modulation is essential for the protection and the prevention of the rejection is happening. As I mentioned to you yesterday, the last talk on Thursday, I think, it's very similar to what happened with the tumors. The tumor is secreting a lot of factors who are educating the immune cells within the tumor in order to prevent the rejection. And we think similar is happening with the placenta. The placenta, the trophoblast, similar as cancer cells, are secreting a lot of factors that are capable of modifying the phenotype of the immune cells that will be present at the implantation site. So the questions, and again, there is so many studies already done in that area by multiple groups, but I think now the questions that are pending is what are the factors? What exactly are those mediators of this balance? And of course, when the balance is established, we know that exists pregnancy complications. So when the balance is established, what are the external signals or the that will induce, the external signals that will break this balance and will be the cause of pregnancy complications such as miscarriages, preeclampsia, and even preterm birth. So extremely important to understand the factors that are regulating the balance in order to understand and prevent the pregnancy complications. So what I mentioned to you is that pregnancy is a crosstalk between the fetus and the maternal immune system. Yeah. And the more and more we study, we see that the embryo, the placenta, plays a critical role on this balance. So it's not one side who's really controlling the whole thing. It's not only the maternal side who's controlling, it's the fetal side also who is sending the signals, communicating with the maternal side. And as a result of this communication, the outcome is this perfect balance. So we have been trying to understand how the trophoblast, how the placenta is able to communicate with the maternal immune cells. And in order to do that, we found that the placenta really is an immune regulatory organ. And what do I mean is an immune regulatory organ? Because similar like any other immune cells, the trophoblast express PRRs, such as the toll-like receptors. The PRRs are specific receptors in the innate immune cells that recognize proteins released either by dying cells, the intrinsic organs, or proteins that are released by foreign organisms such as bacteria, viruses, and so on. And this capacity of recognition allows then the activation of first of the innate immune system and then of the adaptive immune system. As I mentioned, the placenta, the trophoblast more specifically, express all the toll-like receptors that we know, from 1 until 13, both in mice as well as in the humans. As you may know, and just a quick summary, there is the extracellular receptors, TOLR 1, 2, and 4, and there is the intracellular receptors such as TLR3, TLR8, TLR9. The extracellular will recognize soluble factors such as LPS, peptidoglycan, and so on. The intracellular will recognize when the microorganisms are inside of the cell, such as parasites or viruses. In any way, the recognition, either extracellular or intracellular, will lead to the activation of an intracellular cascade and the gene expression of inflammatory cytokines, or as we saw also, the expression of type 1 interferons. All of them are secreted with two objectives. One is to modulate the microenvironment, and second, to control or communicate the innate immune system. So the immune modulation that we find is not only the recognition, as I say, of external antigens, there is a close communication with bacterial products, the role of the microbiome. I don't think we will have time to discuss the growing field of the role of the microbiome in the communication with the trophoblast. Maybe I will touch a little in the last lecture, but as a result of this communication, there are factors secreted by the trophoblast who will be talking to all the immune cells present at the implantation site. So and in addition to the immune regulation, there is also the production of factors that will protect the placenta against viral infections. And again, that is a subject that is extremely important in these days, especially because we're dealing with coronavirus, with SARS-CoV-2, and many other viruses that will be coming in the future. And pregnant women will always be in a special group that may be target of viruses. So it's extremely important to understand how the placenta deals with viral infections. And again, I will try to cover that in the last lecture. So the immune regulation. We know now very well that factors secreted by the placenta are regulating T regulatory cells, are regulating NK cells, stroma cells in the seawater. I have some nice data about that. We'll see if I have time to show you. Is regulating TCD8 positive cells, Th17, and of course the macrophages, which is the subject that we're going to discuss today. All that modulation, again, is to protect, maintain the tolerance, but also to protect against bacterial and viral infections. So how the placenta will function as immune regulator? There are at least 3 stages. One is the recruitment. So the placenta has the capacity to recruit immune cells. The second aspect is in education. The factors secreted by the placenta are capable or induce the differentiation into these unique populations of immune cells within the deciduum, including the NK cells, what discussed already, the T regulatory cells, and what we will discuss today a little more is about the macrophages. So if we talk about the macrophages and the regulation of the maternal-fetal interface. So let's talk, what are the macrophages? This is a unique immune cell population. They exist in every single organ in our body. And within each organ, it has unique characteristics that led for many years to think that they are coming from different origins, because the macrophages in the liver are different from the macrophages in the brain, or the macrophages in the placenta, or in the uterus in general. Now we know that they come from the same origin. They come from these multipotential hematopoietic cells who are in the bone marrow. They give origin to the myeloid progenitor, and this myeloid progenitor differentiates into the myeloblast. And the myeloblast then goes to the circulation as a monocyte. So let me remind you this. There is no macrophages in peripheral blood. Yeah. So that is a classical mistake of people who are not in the field. Macrophages only exist when the monocytes, the only type of cells who are in the peripheral blood, when these monocytes migrate into a tissue and they will go undergo differentiation, then they become resident macrophages, or they can also differentiate into the dendritic cells. Let me make a small note here, because again, for many years, we thought that all the macrophages within the tissues were originated from these monocytes that migrate into the tissue. Now we know that that is true, but it's partially true. So in early stages of differentiation, these myeloblast cells, at the time of fetal development, they will also go into the tissues. And when they go into the tissues, they will create what is called the original tissue, or is like a tissue backup, or the specific tissue originated macrophages. So they exist in an undifferentiated stage within the tissue, and they will remain there until there are specific stimuli that will induce their expansion. But in the majority of the cases, when there is an immune response or changes in the microenvironment, you have a strong component of these migratory monocytes that will differentiate into tissue macrophages. So they are the main source of the tissue macrophages. But again, there is a component that is already established at the time of fetal development. So there is a lot of heterogeneity and plasticity in macrophages, And this is a nice review by Professor Liao's team in Frontiers Immunology, showing a summary of what is a very simplistic, but a well-defined two types of macrophages that exist in tissues. One is assigned as the M1 phenotype, that is the pro-inflammatory. You have a range between this M1 going to the M2 macrophages, which is an anti-inflammatory. And the M2 are also subdivided now in 4 different subclasses, which I don't have time to go through them. And each subclass has a more specific role. For example, the M2B has a role to support tissue growth, repair, and so on. The other ones, the M2C, has a more phagocytic capacity. So the subdivisions are based on the dominant function of these macrophages. But for this lecture and this course, we will use these 2 terminologies, M1 as a pro-inflammatory and M2 as an anti-inflammatory. As we mentioned, macrophages are present in the deciduum, and they are around 20%, 20 to 27% of the cells, of the immune cells present in the deciduum. They are extremely important during pregnancy. They are important in tissue renewal during the placentation and the phagocytosis of apoptotic cells. They're extremely important in spiral artery remodeling. I'll show you a picture about that. And also they are important in the integral part in the cascade of events that is going to happen during labor. They migrate into the myometrium and they initiate in many ways, or they help the changes that will be responsible for labor. A critical role of these macrophages is to maintain this balance between tolerance and the activation of pro-inflammatory responses that will lead to a rejection. They maintain the microenvironment at the implantation site that will fit to the stages of development. And I want you to remember that. They will maintain the microenvironment that is adequate for the development. And we will see what is the meaning of that. Maintaining a microenvironment that fits the development. As I mentioned, they are important in tissue homeostasis and repair. They are cleaning all the dying cells, especially in the early implantation, and they protect the fetus against invading pathogens. Pathogens coming from the blood of the mother, or pathogen is coming from the lower reproductive tract through the vagina and the uterus. They are the first barrier preventing. When we look at the presence of decidual cells, presence of decidual macrophages, it's fascinating to see the physical contact or the physical relationship that these macrophages have with the trophoblast. In these figures, what I'm showing you is the brown is the macrophages, and these blue cells are the trophoblast. And you can see how close and interacting they are, the trophoblast with the macrophages. An interesting aspect is that I mentioned to you they play an important role in the spiral arteries remodeling. So here you see a spiral artery. When we stain it for macrophages, we use in CD68, you see that the macrophages are here, and they are behind the trophoblast, here CK7. The trophoblasts are reaching the spiral arteries, and the macrophages are going behind them, cleaning the place, secreting factors that are extremely important for allowing the macrophage to migrate, to penetrate, reach the spiral arteries, and replace the endothelial cells. They are working together in order to reach the common objective, that is ensuring the communication between the maternal blood and the fetal blood. Again, we talk about the stages of inflammation and anti-inflammation in pregnancy. And in this graphic also from Professor Liao, they show very nice a correlation between the different inflammatory stages of the pregnancy with the stages of the macrophages. So now we know very well that in the pre-implantation, as well as the early implantation, the dominant macrophages is the M1. As the whole microenvironment of the decidua and the implantation site starts switching towards an anti-inflammatory, the macrophages will also change and they will become M2. And here comes the question, are the macrophages responsible by the changes, or the changes are inducing the switch or the polarization into macrophages from the M1 and M2? And they will remain as an M2 for the rest of the pregnancy until the end. And at the end, we will see a migration of M1 macrophages or recruitment of M1 macrophages into the muscle layer of the uterus and responsible of supporting the process of parturition. If those changes will not happen, so the normal pregnancy, you have a balance between the M1 and the M2, especially going to the spiral arteries. As I showed you before, we know that they are mainly already M2 macrophages helping the trophoblast. So in the normal pregnancy, you have the switch into the M2, but if that switch is incomplete and you have still abundance of M1, I have a picture that I forgot to put it here, you will see that the same macrophages, now they behave different. And instead of going behind the trophoblast, they go ahead of the trophoblast, and they really become a barrier preventing the trophoblast to reach the spiral arteries. So a lack of shift between the M1 to the M2 will lead to abnormal implantation, including abnormal spiral arteries transformation. In other words, the interaction between the trophoblast and the macrophage will determine the functional, they will determine the functional characteristics of these cells. So let's now go and try to understand how this interaction is happening. So I mentioned to you, the trophoblast is able to sense, is able to secrete factors that may recruit those immune cells and also educate. How is this process happening with the trophoblast? Sorry, with the macrophages. Are the trophoblasts recruiting monocytes? Are the trophoblasts educating those monocytes in order to become macrophages? Those are the questions that we wanted to understand. So let's start with the question, have the trophoblasts the capacity to recruit monocytes into the implantation site? In order to answer this question, we develop in vitro models that will mimic the placenta or the early stages of placental formation. And what we do is we have in the Petri dish a model that forms villi, exactly like when you see the villi in the first trimester placenta. Let me just show you some nice pictures of those villi. Here you see they form nice network within the structure that this three-dimensional structure that we use. And these three-dimensional structures are communicating with multiple cells, including the stroma cells. And using this, here is a nice view of these villi that you can see here. They are sending projections. As I mentioned to you, they have the capacity to secrete a lot of factors. So we wanted to know whether these villi, these cytotrophoblasts, will be able to communicate with monocytes in the peripheral blood. So what we did is we took monocytes isolated from peripheral blood. They are CD14 positive. We put them in this three-dimensional in vitro system. In the top of the three-dimensional, while the trophoblasts are in the bottom within this matrix gel. And then we monitor how they are moving. So again, if there is no trophoblast cells, these immune cells, they clump in the surface of the model. They don't move, they remain there. However, if we have these trophoblast cells, these projections, these villi, again, let me guide you in this picture. This here in the right corner is the top of the well, and here in the left corner is the bottom. So we're looking at three-dimensional image, and it's because of the focus. If we put the focus now to the bottom with the trophoblast cells, you can see this gradient of cells who are moving from the bottom all the way, from the top all the way to the bottom, and they will reach where is the trophoblast cells. And when they reach the trophoblast cells, they surround these projections made of trophoblast cells, and they will sit around these cells and will start undergoing a process of differentiation. They can remain there for months, and it's interesting because if we have these macrophages now within the system, they are continuously remodeling and cleaning, and these cultures can be maintained for a long, long time. Longer if in cases where we don't put these cells. We quantify this migration, and we show that when there is trophoblast in the bottom, there is a significant increase in terms of monocyte migration to both in 24 hours and 48 hours. So clearly, there are factors produced by the trophoblast who are recruiting those peripheral monocytes into their environment, and then they will do the process of differentiation. So now we knew that at least morphologically, when the CD14 cells will reach the vicinity or will reach the microenvironment when the trophoblast cells are differentiated, the question was what type of lineage this factors secreted by the trophoblast will push the monocytes to differentiate. So again, we wanted to know, just going in a very simplistic way, whether the trophoblast will induce into an M1 or into an M2, either into pro-inflammatory or into an anti-inflammatory. That's going to the immune education. So that is the question, is this going to be into the M1 or M2? And what are the characteristics of these macrophages who are in the deceiver? You see them here with this is extravillous trophoblast, as I showed you before, in the close proximity of these macrophages. Are these macrophages M1 or M2? So to study this, again, we have to go in vitro, and we develop a system of communication between trophoblast and monocytes using the conditioned media secreted by trophoblast cells. And then we will evaluate the differentiated macrophages by flow cytometry using markers such as CD14, that is a general marker for monocytes and decidual macrophages, CD86 for M1, CD206 for M2, and CD16 for differentiation marker. So again, what is the model? We isolate CD14 positive cells from the peripheral blood. We then expose them to soluble factors coming from the trophoblast or cytokines. And then we collect those differentiated cells and we evaluate it by flow cytometry, or we look function, especially pathogen recognition or response to pathogens, tissue repair, and phagocytic capacity. So this is an example of the blood that we will receive. We see the three populations: lymphocytes, monocytes, and granulocytes. We will isolate these monocytes using CD14 magnetic beads. And here you have the CD14 positive cells isolated from the blood, the classical monocytes. Then we will expose these monocytes to the conditioned media secreted by the trophoblast, and then we will see how they change. One of the first things that you can see is that the monocytes are homogeneous population. Again, the small cells in this quadrant. But when we expose to conditioned media from the trophoblast, they change morphological. They become bigger, more granular, classical morphology of a macrophage. Interesting, they don't lose CD14. Again, this is unique to the decidual macrophages because when CD14 monocytes goes to other tissues, they lose CD14. And again, that is a characteristic of macrophage, but not in the decidual. In the decidual, they will maintain the CD14. We look now to additional markers. Again, monocytes, peripheral monocytes. The majority of the peripheral monocytes are CD16 negative. We know that there is a very small population, around 4 to 5% of CD16, CD14 positive monocytes. But the majority, as I say, they are negative. In the presence of factors from the trophoblast, they become CD16 positive, CD14, CD16 positive. So we did a lot of markers. This is a summary of the final characterization. In the presence of factors secreted by the trophoblast, the monocytes become CD14 positive, CD16 positive, CD206 positive, and CD86 negative, suggesting that in the presence of factors from the placenta, they become the classical decidual cells that is more into the M2, is more into the M2 anti-inflammatory macrophages. We look at the functions, for example, phagocytosis. So we differentiate also CD14 positive monocytes with GM-CSF, a chemokine that will differentiate them into M1. When we look which one of the two have a higher capacity of phagocytosis, the macrophages, the decidual macrophages, or macrophages induce differentiation by trophoblast cells, they have a high capacity of phagocytosis. And you can see in this movie, I hope it will go. See, I think I stopped it. This is a macrophage who differentiated by the trophoblast that has acquired a huge capacity of phagocytosis, is becoming very fat because it's engulfing dying cells. And then it will process and start producing a lot of anti-inflammatory cytokines. A very obese macrophage that is cleaning this environment of apoptotic cells. So we will see also about their function when they are cleaning. Do they have a capacity to repair? So we took a model of a wound repair where we do a wound in stroma cells in culture, and you see this is the wound. We expose those stroma cells to the factors coming from these decidual macrophages or from the peripheral blood monocytes and M1 macrophages, and you see how they repair. Let me show you the quantification just to make, there is a movie here I don't think you can see because of the background, but in the presence of decidual macrophages, or what we call the trophoblast educated macrophages, which is this blue line, this is the time that it takes to repair. As you can see, the factors secreted by these trophoblast educated macrophages, they repair within 12 hours. Just in media, the repair, of course, a cell repair will take around 16 hours. But in the presence of M1 macrophages induced by the differentiation of GM-CSF, the time of repair is delayed significantly. So M1 macrophages are not good for repair. They are more for protection against infections. On the other hand, the trophoblast educated macrophages, they have a high capacity of repair. How are we doing with time? Good. So now we know that the trophoblast is able to recruit monocytes, and upon recruitment, they have the capacity to differentiate them into residual macrophages. So we have been able to recreate in vitro what is happening in vivo. Because again, we have done arrays of those trophoblast-educated macrophages, and they match perfectly with arrays that people have done of macrophages isolated from the deciduum. So they recruit monocytes, they induce them the differentiation into an M2 anti-inflammatory macrophage. So our next objective was to identify what are the factors that the trophoblast is secreting that is responsible for this conversion, for this differentiation. Again, the question, we know that the trophoblast is secreting factors that are inducing the differentiation of the monocytes. What are those factors that will lead to the polarization of M2 macrophages? So again, in collaboration with Professor Liao, we were looking at the PD-L1 signal. And we know that the receptor for PD-1 is present in peripheral monocytes. What we didn't know is whether the PD-L1 is secreted by the placenta and the trophoblast. I'm not going to go in details about this because Professor Joaquim did a fantastic job explaining to you what is the PD-1 and PD-L1 pathway. So I'm going to skip this. Similar as Professor Joaquim showed to you, it has been reported that in the peripheral blood is detectable the presence of PD-L1, and I think that was the origin of this slide. So PD-L1, as you know very well, exists in a membranal form, but also exists as a soluble form or secreted form. And that secreted form is detected in the peripheral blood of a pregnant woman. So we wanted to know where that PD-L1 is coming from, and second, whether that PD-L1 be responsible for the differentiation of these macrophages. So the first question, where is coming from? Do the trophoblasts express PD-L1? So we took trophoblast cells, again, our cell lines, as well as first trimester trophoblast cells isolated from first trimester placentas, and we look at the expression of PD-L1 by Western blot. So this is the whole lysis. And we can see that PD-L1 is expressed in the trophoblast, and its expression, it doesn't change on time. Again, this is whole lysis of the trophoblast. However, what we did also is we took the supernatant of those trophoblast cells. Again, both cell lines as well as first trimester trophoblast cells. We collect those supernatants and we tested for the presence of soluble PD-L1. And here is the finding. We do detect secreted soluble PD-L1, but interestingly, the amount of soluble PD-L1 increased in the supernatant with time, suggesting that the trophoblast is continuously producing PD-L1, soluble PD-L1, and is secreted into the supernatant in the case of in vitro, but it will go to the peripheral blood, we'll say is secreted into the maternal circulation. Again, soluble PD-L1 is constitutively expressed, is constitutively secreted. This is the trophoblast cell line. This is the primary culture. You can see that is increased during time. So I will show you some data about the maternal circulation at the end, just to keep the focus of this. So we know now that the trophoblast is secreting PD-L1. And we know it's present in that condition medium. So the question that we had was, is the PD-L1 that is secreted by the trophoblast responsible of inducing the differentiation of the macrophage. In order to answer this question, we did the following experiment. We took the conditioned media from trophoblasts, the soluble factors, and we exposed to monocytes who were incubated in the presence of an antibody that is blocking PD-1. So an antibody that binds to PD-1 and consequently will prevent the binding of the soluble PD-L1 into the receptor. So it is a blocking antibody. And in this model, then, we define what happened to the process of differentiation. So let me rephrase now the different groups that we use. So we have peripheral blood monocytes exposed to GM-CSF, which will induce the differentiation into an M1 macrophage. We will take the same monocytes, expose them into trophoblast conditioned media, which I already showed you, induce a phenotype that is an M2 decidual-like monocytes. And now, to the conditioned media, we add an anti-PD-1 blocker. And then we see what happened to the differentiation. So again, we use the same thing, the markers that I mentioned to you. So let's go one by one. When we take GM-CSF differentiated macrophages, we see that they are CD86 positive. Again, that means confirming that they became M1 macrophages. When we take now the trophoblast educated macrophages, we see that they are CD86 negative. Again, showing that induce the differentiation into an M2 macrophage. Now, if we have the same condition, but in the presence of a PD-1 blocker, we look what type of differentiation, and we found that they became CD86 positive. So although there were many other factors present in this conditioned media, by blocking PD-1, they maintain the CD86 expression, which is a characteristic of M1 macrophage. In other words, by blocking the PD-1 or the PD-L1, PD-1 pathway, we have reversed this M2 macrophage into an M1-like macrophage. So PD-1 is extremely important in soluble PD-1 produced by trophoblast cells, produced by the placenta, are essential for the differentiation of monocytes, or at least also M1 macrophages, into an M2. And what as I mentioned to you before, since the placenta seems to be producing early soluble PD-1, we went and we looked in the blood of the mother in the very early stages of the pregnancy, when the placenta is already, when the trophoblasts start invading the decidua, and is the process of the formation of the placenta, the placentation. And indeed, what we found is that PD-L1 already between 4 and 5 weeks of pregnancy is detectable in high levels in the peripheral blood of the mother, and it continues increasing during the first trimester of the pregnancy, suggesting that as the placenta grows, as the placenta is formed within the decidua and continues producing factors in the communication with the mother, with the maternal immune system, this soluble PD-1 seems to be extremely important in polarizing the endometrial macrophages into an M2 phenotype. So we're doing a good time, so we are almost done. Let me try to summarize what I have shown you in the last 45 minutes, and then we will have time for questions, if you have any questions. So we know now that at the time of implantation, we have a lot of M1 macrophages. And I show you also in the talk when we discuss the subject of the inflammation and implantation. They are important for communicating with the residual cells and creating this microenvironment that will help the trophoblast to invade. Once the trophoblast invades and there is the need of initiation of the repair process, the trophoblast is secreting a lot of soluble PD-L1. This PD-L1 will bind to its receptor, both in M1 macrophage as well as newly recruited monocytes, and will induce the differentiation into the M2 macrophages. And these two macrophages will start secreting factors who will help further the migration and invasion of the trophoblast, but also will initiate the process of tissue repair and the cleanup as the trophoblast invades. In addition, we won't have time this time, these M2 macrophages will continue producing a very important factor that is interferon beta, which hopefully we will discuss on Wednesday, tomorrow, but it's an area that still we are learning a lot. So I know this is a little, it's a small, the figure, and I apologize, but I want to put you like a general picture of what's happening in the different stages that we have discussed. So the first one is what we discussed at the time of implantation. Yeah? The macrophage produce TNF-alpha, acts on the residual cells. This is M1 macrophages, acts in the residual cells, and these residual cells create this gradient who is leading for the trophoblast invasion, migration and invasion. Once the trophoblast has invaded and is established, immediately they start secreting soluble PD-L1 who will transform these M1 macrophages into M2 macrophages. We also know, and this is a paper that we just published recently, that these trophoblasts are secreting HCG. I mean, we didn't polarize it. HCG secreted by this trophoblast are acting on the residual cells and suppressing the expression of inflammatory cytokines. So two roles. One is transforming the M1 macrophages into M2 macrophages, and second, suppressing inflammatory cytokines produced by these stroma cells or decidual cells. They help the conversion into full decidual. My apologies. The third component in progress is the new findings that we have that interferon beta expression by the trophoblast plays a critical role in the modulation of many of those immune cells. We know that if we knock down the trophoblast, the interferon beta in the trophoblast, much of this polarization is not going to happen. And again, this is work in progress. I hope in the next course, I will give you more details of what's going here. But this is the communication, this is the balance that is established at the implantation site and is by multiple players. But the main aspect that I want to leave you here is that the success of the pregnancy is not just how the mother is responding, what it's doing, and so no, it's really how it's communicating with the trophoblast, with the placenta, the fetal signals. And the success of the pregnancy is based on the proper communication between these two components. If something goes wrong, the same trophoblast will send different signals, and then the mother will change its differentiation and will induce the rejection. And with that, I complete, and I will be happy to answer any question that you may have. Thank you.
Thank you, Dr. Kiu. So, any questions for the students? Okay. Hu Hao, please.
So let me read the question. So if somebody wants to read by yourself, it's okay. If not, feel free to write and I will read the question. So you mentioned M1 dominance may lead to adverse pregnancy outcomes from spontaneous abortion. Thus, we know the mechanism of macrophage in the process of recurrent spontaneous abortion. Yeah, so that is a very good question. If you remember in the lecture of the implantation that I mentioned to you, that inflammation is necessary for trophoblast from the blastocyst attachment and trophoblast migration. Yeah. And that is led by M1 macrophages. And I think the evidence are very strong supporting that. Now, a critical step I mentioned to you in order to maintain the pregnancy is the shift from M1 into M2. That is, even from an evolutionary point, the critical step. The maintenance of the placenta, the maintenance of the fetus, the maintenance of the formation of the placenta is based on that shift. A source of differentiation, a source of the change from from inflammatory into anti-inflammatory are macrophage cells. I don't want to say it's the only one, but it's a very important source. So what I'm proposing to you, or what I'm discussing here, is that once the trophoblast has invaded and start producing HCG, PD-1, soluble PD-L1, TGF-beta, and so on, those factors shift or promote the polarization of those M1 macrophages who already have done their function into the M2, and the pregnancy continues. But if they don't respond, if because of additional stimuli like infection, if they remain as an M1 macrophages, you're mimicking the end of the pregnancy, parturition, that I told you. Parturition is M1. So they will maintain the pro-inflammatory environment, and at the end, other cells, NK cells, dendritic cells, cytolytic T cells, they will migrate and they are the responsible of terminating the pregnancy. So I am not saying, I'm not proposing that macrophages are the ones who are rejecting. Macrophages, by remaining as M1, will maintain the activation of cells, such as cytolytic T cells or activated NK cells, who at the end will be the responsibles of the rejection. I hope I answered your question. The second question is, do we have a medical measure to protect pregnancy by regulating M1 and M2? Brilliant question. The findings of PD-L1 and PD-1 suggest that that could be a potential approach. If we see that the pro-inflammatory condition continues and PD-L1 expression in the blood is low, potentially we could do delivery of soluble PD-L1. That is something that is in work. Or HCG. HCG is one of the best anti-inflammatories. And there is evidence that HCG can also contribute to macrophage differentiation. Again, I don't have any specific therapy, and I don't know if Dr. Joaquin would like to also help with this, but I think by these findings, we can now, it's opening a door of opportunities to develop new ways to monitor those patients, but more important, as you question, ways how to intervene and help those patients with recurrent abortion. And I don't know, Dr. Joaquin, if you want to add something about therapy in this.
Still, the therapy is under development, so it's gonna be hard. But I just got the, we're working on to get the pregnancy outcome data from those NIH trial of PD-1 and PD-L1 blocker study.
Oh, yeah.
Yeah, so that's just now going through some kind of review process now. So hopefully it can be published in some of the oncology journal, because those drugs are utilized for oncology cases. And some of the patients were on those, but kind of compared to the whatever we expected from animal study, those drugs really did not affect that much to the pregnancy outcome, although the number is very small. And I pretty much believe we're not going to get any big data. So I think this soluble PD-1, PD-L1 study will give us a real good clue about this kind of treatment. So it is all we need to study more, but that's the current preliminary, small study. But I'm pretty sure we're not going to get any big one for a long time. But I'm very pleased to see this soluble PD-1, PD-L1 studies are coming out, and thank you for doing this for Dr. Gilmore and Dr. Liao.
Thank you, Joanne. That is a very good point. And again, we have to keep our mind also when we review, when that data comes out, to remember that the timing is everything. So
right.
When the blocker of PD-1 and when is the function?
Yeah, so only a few patients were exposed during the first trimester. Some of them are exposed way before the pregnancy. So, I mean, this is just kind of happened to be exposed. It's not anything kind of clinical trial. And since there's a lot of animal studies that are talking about possible harmful effect of those drugs, so you cannot really do that. But this patient happened to be, but I think one of the other way to explain this is kind of a soluble PD-1 and PD-L1, because these studies really don't have any measurement of those, and maybe that's why we're seeing some kind of different outcome than what we expected to see. Actually, the trophoblast role is there, which we're completely neglecting it. In some sense, and maybe that's why this drug treatment to block the PD-1 and PD-L1 really did not, cause too much trouble in human pregnancy so far.
Exactly.
Mm-hmm.
Thank you, Joanne.
You're welcome. And since I'm on the podium, I just want to ask you, because when I had a PD-1, PD-L1 data presented, one of the students talking about the soluble PD-1, PD-L1 and co-stimulatory molecules. I don't know whether he's still here. I don't remember his name, but he had that question about how the soluble PD-1 and PD-L1 is actually stimulating the cells. That was the question, as I recall. So if you can explain to them, that'll be great.
Fantastic. So, if I understand, he was asking about the molecular mechanism, or
right, molecular mechanism of soluble PD-1 and PD-L1.
Okay, perfect. Yes. So remember, the soluble PD-L1, similar to HUST ligand or to any of these checkpoints, what they induce is in intracellular pathways that will activate gene expression, and they are classical receptors. So when we call them the checkpoints, the majority of the information that you get is just binding to the receptor. But there is a lot of work now looking at the intracellular cascade. PD-1 induced the activation of a specific signaling pathways that will induce the expression of receptors associated with inflammatory characteristics. Yeah? So all anti-inflammatory characteristics, of anti-inflammatory characteristics. So let's put it in the T cells. When the PD-L1 will bind to its receptor PD-1, will activate genes who will promote the differentiation into a T regulatory cell, or will induce the differentiation into a dormant, or no, it's not a memory cell, into a dormant T cells. There are works also showing that they induce the expression of many of the genes who control exhaustion. Again, I don't have a figure to show you here all the intracellular pathways, but remember, this is receptors who control multiple genes associated with cell differentiation. So again, when the PD-L1 binds to the PD-1, activates the expression of genes who control cells differentiation. The same we think is happening into macrophages. Will induce the expression of many genes who push the macrophage into an M2 differentiation. And I think that as the work induce also the expression of metabolic genes who are changing the metabolic function of immune cells, that also are important in the process of immune cells differentiation. It's an area that is extremely hot. And again, I don't understand much about metabolism. We did an array comparing cells treated with different checkpoints. And one of the main pathways who are activated with these checkpoints, not only macrophages and T regulatory cells and so on, is metabolic pathways. And I think Professor Liao has some data also in terms of metabolic pathways affected by the PD-L1. So they are receptors who control transcription of genes associated with cell differentiation. And again, I apologize very quickly, just because of time. I hope I'm able to answer your question. Let me read another question that we have here. Macrophage plays an important role in maternal-fetal interface. I would like to know how can abnormal macrophages lead to pregnancy complications? What kind of pathological pregnancy will occur? The message that I would like to convey to you, there is no good macrophages and there are not bad macrophages. All are good. The problem is timing and location. So if you don't have M1 macrophages early in pregnancy, there will not be implantation. So you have implantation failure. Now, if you have excessive M1 macrophages, excessive inflammation, you will have a rejection. If still the change is not complete, because I'll show you with the PD-L1 blocking, we don't completely shift into M1. They still maintain some M2. So they become a chimeric, a mixture of M1 and M2. What we think is when there is no full polarization, these mixed macrophages don't help properly. So they neither will help the trophoblast to invade, neither will induce a rejection. So you have pregnancy complications of an unhappy marriage that I like to call, that will be preeclampsia. And we know that these non-terminally differentiated macrophages are responsible of the normal spiral arteries transformation. Yeah. So again, what is the degree of the differentiation? What is the time of the differentiation? Because based on those aspects, the function, the specific function in a specific times will be affected. Do we have questions? Let's see. There is another question here. Trophoblasts contain genetic material from the father, and healthy trophoblasts is very important to maintain the immune balance at the maternal-fetal interface. When father has genetic problems, miscarriage is common. I want to know whether trophoblasts cannot induce macrophage differentiation is the reason for miscarriage. Thank you. Yes, yes, absolutely. We cannot blame everything on the mother. There is a paternal imprinting, which is extremely important in the placenta, extremely important. Yeah. And again, this is data that is just coming. I cannot mention more because it's incomplete, but I hope soon I will be able to show you. Abnormal paternal imprinting leads to abnormal expression of those factors who will either maintain a pro-inflammatory environment or exacerbate the inflammation. So absolutely, whoever, I think is, whoever asked the question, yes, paternal imprinting in the placenta is critical, and an abnormal paternal imprinting will lead to an abnormal development of the placenta and consequently abnormal development of the microenvironment. Have here at the Mod Center a group of people who are studying the paternal imprinting, the paternal contribution, also how the environment affects the sperm, and then the consequences of that sperm on the formation of the placenta and therefore the regulation of the microenvironment. So excellent question, thank you. Is there any macrophage involved in the pathogenesis of preeclampsia? Yes, absolutely. Multiple groups have shown, and including our work in, I think it was in 2004, we took samples from the basal plate of normal pregnancies that have late terminations and preeclampsia patients. And I apologize, I should have put those pictures here, but we show that when you're looking at the spiral arteries of the preeclampsia, the spiral arteries were surrounded by macrophages, totally surrounded by macrophages. And the stroma and the trophoblast cells are around trying to reach the spiral arteries. And the macrophages in these cases, in preeclampsia, they form like a wall. And the trophoblast cannot reach the spiral arteries. So abnormal differentiation of macrophages is the cause that they behave as a wall between the spiral artery and the trophoblast, which is one of the causes of preeclampsia, abnormal spiral arteries difference transformation. The next, from the lecture we have learned that trophoblast cells play an important role in inducing differentiated macrophages. So I wondered whether it can be applied to explain the mechanism of trophoblastic tumors. Absolutely, absolutely. And that is the reason I always compare the trophoblasts with the tumors. Remember, the trophoblasts form a type of tumor that is the choriocarcinoma. Yeah? And the choriocarcinoma invades the uterus and transform many of those macrophages, and those macrophages are extremely, they become, it's called an M2C type, which is the classical macrophages that you find in tumors. And they are invasive macrophages. So they teach the trophoblasts how to further invade the tissues, and they never stop, and they invade the muscle, and they propagate in the peritoneal cavity and so on. So yes, and that's what we see also in other type of tumors. Is PD-L1, where is it? Is PD-L1 completely secreted by trophoblasts? Are other placental tissues involved in the secretion of soluble PD-L1? Is the efficacy soluble consistent with membrane receptor PD-L1? Yeah. So we look at the stroma cells to see if they secrete PD-L1. We did not find. The main cells for producing detectable amount of soluble PD-L1 is the trophoblast. Whether the membranal PD-L1 plays a role, I don't have an answer. We did some co-culture, but it's very difficult to see, to differentiate whether there is the membrane or the soluble. So I think, again, that is a hypothesis. I think that both are important. The soluble, why the soluble? Why do you need a soluble? Because in pregnancy, the communication with the maternal environment and the maternal immune system is not only cell to cell. That's what it makes the difference in the mammalian and the human, is at the implantation site, which is a big site, but also in the maternal circulation. And I think Dr. Joaquin brought a fantastic point. Yeah? The placenta is swimming in the blood of the mother. So it's not the old concept that the pregnancy is an isolated condition. The pregnancy is happening in the endometrium, but it's communicating with the mother in the whole body of the mother. The cells of the fetus are going to the mother. The mother cells are going to the fetus. So the factors secreted by the mother are going to have an impact in the placenta, but at the same way, the factors secreted by the placenta and the fetus are going to have an impact on the mother. So the soluble is extremely important for a more systemic general, but I cannot deny or say or reduce the importance of a possible cell-to-cell contact. I show you pictures of how close the invading the extravilous trophoblasts are with the macrophages. So it is a possibility, and I'm convinced that is the case, that there is also a cell-to-cell interaction. I don't know if there is any other question.
Yes, Professor. No.
Go ahead. Yeah, why don't you read or tell me? Go ahead.
Yes, it's Patrick.
Hi, Patrick.
Yes, I was the one who raised the question about soluble PD-L1. I was just wondering, you know, when we talk about PD-1, PD-L1 pathway with membrane-bound PD-L1, we consider that the binding of PD-1 to PD-L1 is the co-stimulatory signal, because there is first the binding of TCR and MHC molecules, you know. But
Yeah.
my difficulty was about soluble PD-L1, since we won't have the first signal, how it could work. That was my question.
Oh, I see. Another question. Again, it's very important to take in consideration the cell type that is the target. Again, the initial studies about all these checkpoints were associated with T-cell activation, where these co-stimulatory signals play an important role. And it's the same as with the FAST ligand, the TNF-alpha receptor, and so on. As we move outside of the T cells into other cells, there is now clear evidence that they are not functioning only through the co-stimulatory molecules. That is what I tried to explain at the beginning, that in many of the cells who do not have a T cell receptor, they function as an independent receptor that will activate intracellular pathways, transcriptional pathways, and gene expression. So it's very cell specific, but there is not only a single mechanism. Within the T cells, the complex of the T cell receptor is, there is a high degree of complexity on the T cell receptor in order to activate or inhibit that specific T cell. And that is when you correctly pointed out the presence of the additional complex. But no, that's not the case in other cell types. And that is an area that is still, we need to understand better whether there is additional receptors who are helping to activate the PD-1 receptor in other cell types, because it's not only macrophages. The more and more they are finding even in some epithelial cells that express the receptor. What it's doing in those epithelial cells, nobody knows. How it's functioning in those cells, nobody knows. In the macrophages, we know more, we are finding more, but definitely is not associated with T cell receptors or with complexes, because macrophages don't have T cell receptors. Patrick, I answer your question?
Yes,
There's a lot of material for research and investigate. Yeah. Again, we have to recognize the primary work has been done in T cells, and as you correctly pointed out, is associated with the complex, the TCR receptor complex. Yeah. That's not the case in other cells.
Okay, thank you, Professor. But I want to have, I'm curious about something that you say. You said that interferon beta plays a critical role in modulating several processes in trophoblast cells. I will not ask how you reach at that conclusion. What I want to know is to go inside your mind of researcher. How do you select specifically interferon beta to do those experiments, reaching to the conclusion that it plays a great role? Because there is several factors, as you mentioned above.
Okay. I will tell you that in a private conversation. What about? Yeah?
Okay, thank you. I will wait.
Okay. Any other question?
I have one. Hi, Professor. Can you hear me?
Yes, I can hear you.
Oh, okay. Actually, I have two questions. One is about the immunotherapy, about soluble PD-L1 in recurrent pregnancy. Have you compared the concentrations of soluble PD-L1 in between women with normal pregnancy and recurrent pregnancy loss?
So I have a figure that I didn't put you. I can share with you that figure because we look at miscarriages, early miscarriage. The numbers that we have, let me put it this, we did not see a major difference. We couldn't separate completely between the normal levels in the peripheral blood between the healthy pregnancy and the miscarriages. So again, it was different from what I show in my first lecture, where we have a clear separation between the ratio of IL-10 and TNF-alpha, which you can see really a beautiful separation between the healthy and the early miscarriages. PD-L1, soluble PD-L1 alone did not allow us to discriminate between the two conditions. So that is a reason when we published this work of the secreted PD-L1, we did not include in the miscarriages because alone does not function as a marker. Now, we just finished doing another study in combination with other markers, and in PD-L1, soluble PD-L1 in combination with other markers is giving us also a very nice differentiation. Unfortunately, the number of cases that we have is not so big in order to create a good statistics, but we are working on that. Long answer to your question, soluble PD-L1 by itself cannot be used as a marker to predict a miscarriage. And your second question was?
My second question is because you said at the time of embryo implantation, trophoblast do not secrete soluble PD-L1, so it cannot differentiate macrophage 2 differentiation. But at the later time, the increase of PD-L1 can help macrophage differentiating to type 2. But I remember there's a paper published in AJRI. It said the concentration of PD-L1 continually significantly increased during the whole pregnant time. So I'm wondering, at the time of birth, so there are still lots of soluble PD-L1, but macrophage differentiate to type 1. So do you think?
Yeah. I know you're talking about that's correct. So it's interesting. The paper shows that paper in AGRI shows the expression of PD-L1 starting in week 8 of the pregnancy, and they look until delivery. So they see there is no major difference in the early there is no, so increase up to 35 weeks, and then after delivery, there is a little down. So it is interesting, although there is not a major difference once it reaches, let's say, 25 weeks and so on, in the increment of the pregnancy, clearly is all the time secreted into the peripheral blood, and you can detect it in the peripheral blood. So if I understand your question, let me see. What I see of this continuous expression of soluble PD-L1 in the blood is a potential marker for placental function more than other thing else, because it correlates, it beautifully correlates with the growth of the placenta. What are the functions of this soluble PD-L1 later in pregnancy, in the second trimester, in the third trimester, which, by the way, at delivery start coming down? I really don't know. And I think that is something that we will learn, but what Dr. Joaquin was mentioning, these studies of pregnant women who receive anti-PD-L1. And I'm sure they receive that potentially in the second trimester or even in the third trimester. Yeah. So that's what we will learn a little more, what is their function. But again, I see the circulating PD-L1 in later stages as a potential marker for the normal development of the placenta. What it's doing in terms of maintaining the immunological balance within the whole circulation later in pregnancy, that's an excellent question. And I will give you my easier answer. I don't know.
Okay.
That is a very good and very important question. But you know, the whole field is relatively new. So there is a lot of things that we can learn now that we have the tools, and we know it's there. We know it's changing during the pregnancy. We have no evidence of what is acting in different immune cells. There is a lot to know what it's doing in later pregnancy, how it's maintaining those stages. Thank you for the question.
Thank you.
I don't know if there is more questions. Let's see. Professor Liao, there is more questions in the
yeah, one more question is from Qin Danyu. Maybe you can read. Oh, no. She's in the library. Her question is about what the relationship between T cells and macrophages in maternal and fetal immunity.
Good. Okay. So thank you. Also very good question. Remember, macrophages are innate immune cells, and their role is to recognize the microenvironment and send signals to the T cells and tell the T cells, they really translate to the T cells what is happening in the specific microenvironment. So a macrophage who is highly activated towards a pro-inflammatory, it will be producing a lot of cytokines that are responsible of the activation of cytolytic T cells. And that signal will tell them, I am having problems here. You need to come and help me. So if you have an infection, for example, a bacterial infection or a viral infection, the same macrophages who were keeping the pregnancy happy, they are going to switch their polarization. Some of them are becoming M1, stimulated by the virus or the bacteria. And then they will be communicating with the T cells and recruit the T cells, the cytolytic T cells, who will initiate the process of rejection. And the same will be with the NK cells and so on. So the concept that you have to keep in your mind is that the innate and the adaptive immune system are not working separate. The T cells are not working in a different planet or in a different world. All of them are working together. And the main role of these cells, and that's the reason I like these cells, macrophages, the innate immune cells, is because they are the police. They are the controllers. They are the directors of the symphony. They see how the violin is playing and how the guitar or whatever other instrument is playing, and they communicate with them and they tell them how they should tone the song. And the signals from the public is the signals that are coming from the placenta and communicating with the conductor. When they work together, it's a beautiful symphony. If the conductor is giving the bad signals, the symphony will sound terrible. And if the public is not listening and is talking, the conductor will not be able to pass the messages to the symphony. A happy pregnancy is a well-directed symphony. Okay, and with that, I think we will end, yeah? Thank you very much, and I will see you tomorrow for the last lecture.
Yeah, okay, thank you, Dr. Gill, for your wonderful lecture. See you tomorrow.
Thank you so much to all of you.
Yeah.
Thank
Bye-bye.
you. Bye.
Bye. Bye, John.
Bye, John. Let me see, I have to stop recording.